Hello everybody, welcome to Deeksha Online. So we had started discussing the chapter called Sexual Reproduction in Flowering Plants. Now we are going to discuss the second lecture in the same chapter. Now to give you a chapter overview, we have already discussed about the flower, which is a fascinating organ of angiosperms. We have discussed a few pre-fertilization events, that is structures and events associated specifically with, if you remember, the anther. Then we will be discussing about a process called double fertilization. Then moving on to what happens after fertilization of that is between the male and the female gametes, resulting in the formation of zygote, that is formation of a seed, which comes under post-fertilization. And last but not the least, we will be discussing about some unique phenomenon such as apomixis and polyembryony. So this gives you the chapter overview of what you will be learning in this particular chapter. A quick review of what we had done in the previous lecture. Now in the previous lecture we had discussed about flower and its parts, isn't it? So what are the essential parts, what are the non-essential parts? The essential or the reproductive parts, we had classified them into the female part, which is called the gynoecium, and the male part, which is the androecium. Yes or no? Then we had discussed about pre-fertilization structures and even specifically with regard to stamen, where you know that the stamen has a stalk which is called the filament and the anther, and we had seen what you will see if you take a section of the anther. We had seen that there are structures called microsporangia, isn't it? How many microsporangia are there in a typical Dithecus anther? There are 4 microsporangia. We had seen that the microsporangium has a tissue in the middle called the sporogenous tissue, surrounded by 4 wall layers. Okay? Then we had seen what exactly is the process of microsporogenesis, how a cell called microspore is formed, and each of that microspore will transform itself into a 2-celled or a 3-celled pollen grain. We had seen that when the pollen is released from the anther, in 60% of all the angiosperms, it is 2-celled. It has a larger vegetative cell and a smaller generative cell. In the remaining species, it is 3-celled. Okay? So this is a quick review of what you had learned in the previous lecture. Now in this particular lecture, we will be discussing about the female part of the flower. Now what is the female part of the flower? The female part of the flower is referred to as the gynoecium. You all are aware. And the unit of gynoecium is referred to as the pistil or the carpel. Just like in the male part we had called it the androecium. The unit of androecium was the stamen. Similarly, the female part of the flower is referred to as the gynoecium. The unit of gynoecium is the pistil or the carpel. So remember, the gynoecium is what part of the flower? The female reproductive part of the flower, and the units of the gynoecium are referred to as the pistil or the carpel. Okay, so now let us see what a pistil or a carpel looks like. Each pistil will have, we have done this before, so we'll have an expanded part, a platform like structure on top, and then it will have a very elongated neck, isn't it? And then the basal part of the pistil will be swollen like this, and this pistil is located on the expanded part of the flower. What is this expanded part of the flower called? It is called the thalamus, isn't it? So what are the 3 parts of the pistil? This upper expanded part is referred to as the stigma, and this, the narrow region, the neck of the pistil, is referred to as the style, and the basal swollen part is referred to as the ovary. Okay, so what is there inside the ovary? Inside the ovary there is a cavity. The cavity inside the ovary is referred to as the locule, or it is called the ovarian cavity. Clear? So we have now seen that the 3 major parts of the pistil are the stigma, and then there is a stalk, or the neck we can call it rather than the stalk, which is the style, and the basal swollen chamber, which is referred to as the ovary. And inside the ovary there is a cavity that is referred to as the locule or the ovarian cavity. Then at the base of the ovary, this is only purely for diagrammatic purposes. At the base of the ovary, there will be a nutritive tissue. Now, this nutritive tissue is referred to as the placenta. It may be anywhere in the ovary, but I am showing it at the base of the ovary. There is a nutritive tissue that is called the placenta, and attached to the placenta is a structure, and this structure, which is attached through a stalk to the placenta, is referred to as, what is it referred to as? It is referred to as the ovule. The technical term for ovule is the megasporangium. Is that clear to all of you? So sitting inside the ovary, there is a structure. We will discuss this structure in great detail in this chapter, in this particular lecture. This structure which is sitting inside the ovary is referred to as the ovule. The technical term for ovule is the megasporangium. And then you can see that the ovule is attached to a nutritive tissue that I have represented at the base of the ovary, and this nutritive tissue is referred to as the placenta. And what is the technical name of ovule? The technical term for ovule is the megasporangium. Is that clear? So now we have discussed about the structure, the unit of the gynoecium, and this unit of gynoecium that is made up of the stigma, the style, and the ovary is referred to as the pistil. I am repeating again for all of you. Inside the ovary there is a compartment, there is a chamber which is referred to as the locule or the ovarian cavity or the ovarian chamber, and inside that there is a structure which is called the ovule. The technical term for ovule is megasporangium. The ovule has a stalk which is attached to a nutritive structure through which it derives its nutrition, and this nutritive tissue is referred to as the placenta. Is that clear to all of you? So just now we discussed, this is the summary of what we just discussed. Each carpel or pistil has three parts: the stigma, the style, and the ovary. And I told you the ovary encloses a cavity called the ovarian cavity or the locule, and sitting inside the ovary, I told you that there is a structure which is very, very important and our focus in this particular lecture, which is referred to as the ovule or the megasporangium. Okay, keep this in mind. Now, I showed you only one pistil in the previous slide, isn't it? I showed you one stigma, one style, and a basal swollen part, which is referred to as the ovary. So this whole thing which I drew in the previous slide was one carpel or one pistil. So a flower may have a gynoecium which has only one carpel or pistil. Sometimes it may have two or more. It is not necessary that a flower always has only one pistil. It may have two or more pistils, and if there are two or more pistils, they may remain free from each other when the condition is called apocarpus, or they may get fused with each other in a condition that is referred to as the syncarpus. Okay, now for example, let me show you here in a simple way. Imagine that this is the thalamus of the flower. This is purely for your understanding. Okay, so this is the thalamus of the flower. Say for example, I am drawing 1 and 2. So these are how many carpels now? This is a carpel. So let me call it as, we'll use the word pistil. So that is what is routinely used in your NCERT as well. So this is pistil 1 and this is pistil 2. So now see this and tell me, are they fused or not? They are not fused. They are very well independent of each other. This pistil 1 has ovary of its own, it has style of its own, it has stigma of its own. Similarly, the pistil 2 has ovary of its own, it has a style of its own, and it has the stigma of its own. So we call this condition where the two pistils of the flower, to simplify the concept, I have taken only two pistils here, we call it as apocarpus. They are not fused. Now imagine that they fuse together. Now how do you know that they have fused together? So let me try and give you a simplified example here. Let me take another flower which has two pistils, but see how they have fused together. So I'm showing two carpels or two pistils here, but it looks like one, isn't it? It appears like one. They have fused together. So how do you know they have fused together? Now first of all, can you all see there are two stigma here? This is stigma 1 and this is stigma 2. This is one clue to you for you that yes, they have fused together, because here they look separate from each other, but here it is fused together. Another easy way to identify whether they are fused or not is you take a transverse section. So when you take a transverse section of this kind of a pistil or this kind of a gynoecium where two pistils have fused together, you will obviously see inside the ovary. Now imagine you have taken a transverse section of the ovary. Inside the ovary you will see two compartments. Okay, what is this compartment or the chamber inside the ovary called? We called it as the locule or the ovarian chamber. So this is one chamber, the locule 1, and this is another chamber, that is locule 2. If you have two compartments or two chambers inside the ovary, that means it is probably formed by the fusion of two separate pistils which have become one. Okay, so obviously you will find ovules in this chamber also, and you will find ovules in the second chamber also. So in this condition, in this simplified example, I have shown you that two pistils are separate, which is referred to as apocarpus condition. Now what should we call this condition where the two pistils have become fused together? We should call this condition as syncarpus condition. So please bear in your mind, how do you know that they have fused together? The best way to find out is you take the transverse section of the ovary. If you see that there are two locules, in this example I have taken two carpels, so you will obviously see two locules inside the ovary, not a single locule. If it is a single pistil, obviously if you take a transverse section, you will only see one chamber inside or one compartment inside in which you will see the ovule is sitting. But here you are seeing two locules because it is a fusion of two pistils. Is there any other way of telling that this is a fusion? This particular case is a fusion of two pistils? Yes. How many stigma are you seeing in the carpel here? You are seeing one and you are seeing two stigma. So that should tell you that probably there are two carpels which have come together and fused, and this condition where the carpels are fused is referred to as the syncarpus condition. Okay, so I hope it is clear now. The first condition, which is what you see on the left side, is where the two pistils are separate, they are not fused. It is called apocarpus. The second condition, which I have shown on the right side, is where two pistils have fused together and so they appear as one, but when you take a cross section of the ovary, you can see two locules inside, and sometimes you may also see two different stigma which are projecting out from the style. Clear? So that means the carpel have fused, and that condition is referred to as the syncarpus condition. So we now learnt about the condition of gynoecium which has more than one carpel. If it has more than one, for example, I took an example where there are two. If they are free, it's called apocarpus. If they are fused, it is referred to as syncarpus. Okay. So here are some examples. that is in the first example, that is in A, this is actually the ovary, the pistil of hibiscus. See the parts of the pistil. There is a basal part which is called the ovary. There is a narrow neck which is called the style. And see, there are so many stigma. What do you mean? Let us count the number of stigma. 1, 2, 3, 4, 5, 6. Probably it is a fusion of 6 carpels, because generally a carpel has only one stigma. But here, if you see that the stigma has branched itself into 6, or you can see 6 stigma together, then probably it is a fusion. So what type of ovary is it? Is it a gynoecium that is apocarpus or syncarpus? It is a gynoecium which is syncarpus, that is formed by the fusion of pistil. Even though it looks like one pistil, it is actually formed by the fusion of many pistil. Now you answer my question. If you take a transverse section of the ovary of this gynoecium, will you find one locule, or will you find, this is not a rule actually, sometimes even though it is syncarpus you may find only one locule. There are exceptions to that, okay? But for your understanding, you should think and tell me whether you will find one compartment or one chamber inside, or will you find 6 chambers inside, that is 6 locules. I am considering it as 6 because they have shown the number of stigma as 6, only for your understanding, okay? It may not be scientifically accurate based on the flower, but based on this diagram I am considering 6 stigma. So this is a compound pistil which is a syncarpus gynoecium. So you tell me, if you take a section of the ovary, will you find one locule or will you find 6 locules? So you will think about this, okay, and discuss with your concerned teachers. Then the second one is multicarpellary syncarpus. See the ovary here. This ovary doesn't have style at all. It is highly reduced. You can only see the stigma. I am focusing on this diagram right now, the middle one. You can see the stigma, a very, very ornamented stigma, and you can see the basal swollen part which is referred to as the ovary. How do they know that it is syncarpus ovary? Again, if you take a cross section of the ovary, you will see multiple chambers or multiple locules. The number of locules is usually corresponding to the number of pistils which have fused together to form a syncarpus pistil. This is seen in the case of poppy, that is papaver. Then now focusing on the last one, see how many pistils are all there, but have they fused together? They have not fused together. So we call it as multicarpellary. Many carpels or pistil are present. Apocarpus, they are not fused together, and this is seen in the case of Michelia champaka. Okay, this is a flower that is called Michelia champaka. See how clearly you can separate the pistil from here. Each pistil will have its own stigma, will have its own style, and the basal swollen part which is referred to as ovary. So ovary, and then the style is very reduced as you can see, and this upper portion will obviously be the stigma to receive the pollen grain. So each one here represents a pistil or a carpel. Notice they are all separately present. They are not fused together. So as a result, we call this condition as apocarpus pistil or apocarpus gynoecium. Okay, so the first one is syncarpus, the second one is also syncarpus, the third one is, so let me write here, this is syncarpus, this one is also syncarpus, and the last one where the individual pistils are separate from each other, they are not fused, we call it as apocarpus. Okay. Now I told you inside the ovary there is a ovule. Now the number of ovules will vary. Now in all the diagrams have been showing that the basal or the swollen part of the pistil there is only one ovule sitting inside. In all the diagrams I have shown you, it is not necessary that there is only one ovule sitting inside. This is the ovule, isn't it? There are ovaries which have more than one ovule inside. Examples for those ovaries or those angiosperms which have only one ovule in their ovary is wheat, then rice or paddy, and mango. That's why mango has only one seed in it, because usually per ovary there is only one ovule. You all know that the ovule after fertilization, much later, following sexual reproduction stages, it becomes a seed. Okay, so one ovule, how do you know that the plant had one ovule? If the fruit has only one seed. Now if you see fruits like papaya, watermelon, and some of the orchids, you will see that they have many seeds in them. When you say that they have many seeds, that means they have many ovules in their ovary. So remember, the number of ovules may either be one in the ovary or it may be many. Examples you have to learn. Okay. Now coming to the structure of the ovule. Okay, now what is represented here is an inverted diagram. It's the upside down diagram of the ovule. Okay, so I will be drawing the a proper orientation. You can learn it like this also, but for our convenience we will be drawing a diagram with a different orientation of the ovule, and we will be discussing about the parts of the ovule. Okay, now this is how an ovule looks like. What is the technical term for the ovule? The technical term for the ovule is a megasporangium. I told you that a megasporangium or the ovule is located inside the ovary. It has a stalk. The stalk is attached to a nutritive tissue that is called the placenta. Okay, now I will do a diagram first, and then you will understand what this diagram, this picture that is given in the NCERT is all about. Okay, so let us now once again, to give you a clear orientation, you know that inside the ovary there is a nutritive tissue called placenta, and there is a structure attached to it which is referred to as the ovule or the megasporangium. So this is the ovule or the megasporangium. We will be discussing the structure of the ovule or the megasporangium. Okay, so we will be considering this structure. See how it is upright. Okay, so let me now draw the placenta here. This is the placenta to which the ovule is attached, and now I'm showing the stalk of the ovule, and see how the stalk of the body of the ovule is curved, isn't it? See how it takes a 180 degree. This is the stalk. Okay, now let me focus on the body. This stalk of the ovule is referred to as the funicle. Now I'll come to that. Now let me complete the diagram first. Now the body of the ovule, as you can see here, it is covered by a two-layered envelope. See, there is a two-layered structure which encircles the body of the ovule. Now I am showing you the stalk. The stalk attaches to the body of the ovule. This point of attachment, before that let us label the stalk. What is the stalk of the ovule called? It is called the funicle. Okay, and this is the placenta. Where is the placenta present? The placenta is attached to the wall of the ovary. I'm not showing you the ovary here. Okay, so funicle. Now see this point, all of you. This is the point where the funicle attached itself to the body of the ovule, isn't it? Now this point where, or this region where the stalk, this is the stalk. This is the stalk. As you can see, the stalk is attaching itself to the body of the ovule, and this point is referred to as the hilum. Hilum is the region where the funicle attaches to the body of the ovule. Is that clear to all of you? Then inside the ovule there is a main tissue. I'm showing you the tissue now. There is a rounded mass of cells inside the ovule. Let me just show a few cells over here inside the ovule. This is the main mass of cells, and these cells are richly embedded or laden with food reserves, their nutritive tissue. Okay, so this tissue that makes up the body of the ovule, the main part of the ovule. See, all these are cells. And this tissue is referred to as, the nutritive tissue is referred to as the Newillas. What is it called? It is called the Newillas. Remember, the cells of the Newillas are richly provided with reserve food material. Now, can you see two coverings which are surrounding the Newillas, which envelope the Newillas? Now, you can see an outer covering here and you can see an inner covering here. Now, these two layers which encircle the Newillas, these two protective coverings are referred to as integuments. And how many layers are there? There are two integuments. We call such an ovule which has two integuments as bitegmic ovule, because it has two coverings. Okay, there is one envelope inside and there is one envelope outside. There are two layers of coverings around the Newillas, and that is referred to as the integuments. And then the opposite side, this is the base of the ovule. The base of the ovule is referred to as the chalaza. Now I want all of you to notice that the integument is not there at this point. See, there is an opening here. This opening of the ovule, where what is missing? The integument is missing. This is referred to as the micropyle. Very important. So micropyle is where it is considered to be the opening of the ovule, where what is not there? Where there is no integuments. The integuments are the protective coverings. They are missing in this region, the opening of the ovule, which is referred to as the micropyle. Now there's one more thing you should know. Sitting inside the ovule, there is a structure which we will be discussing later in greater detail. So I'm not going to draw that in this diagram. I'm just going to shade it. But there are cells inside it. Right now at this juncture, you don't have to know this particular thing. So sitting inside the ovule, there is a structure, and this structure is referred to as the embryo sac. The embryo sac has egg in it. Because it has the egg, obviously it is a plant or it is a tissue bearing a gamete. So we call this embryo sac also as a female gametophyte. Like how we had called the pollen as the male gametophyte, because pollen produces the male gamete. Now since the embryo sac has egg inside it, and it obviously represents the female generation, and it represents a tissue or a very simplified phase in the life of a plant, which is called a gametophyte. Egg is obviously what? Egg is a gamete. So it is a phyte, a plant. It's a mass of cells which contain egg in them. So it is referred to as the female gametocyte or the embryo sac. Did you understand the structure of the ovule? So I'm repeating again. The ovule has a stalk. What is the stalk of the ovule called? Funicle. The point where the stalk joins the body, the junction between the stalk and the body, hilum. The base of the ovule, chalaza. The main nutritive tissue of the ovule, Newillas. And the coverings of the Newillas, which envelope the Newillas, integuments. How many coverings are there? Two coverings are there. The region where covering is not there, the opening of the ovule, micropyle. Inside the Newillas, there is a structure which represents the female gametophyte because it has egg in it. This structure is called the embryo sac. This type of an ovule which I have drawn here is referred to as the anatropous ovule. Now anatropous ovule is an ovule which is bent on its own side. So here on itself, see how it is bent over here, isn't it? When you draw, you can actually see the stalk, and then it bends like this, and then the micropyle is present on the lower side. So such an ovule which is bent on itself is referred to as the anatropous ovule. Now look at this diagram which I have shown you and see the textbook diagram. It is exactly the same, but it is shown in an upside down manner. You can either draw it like this or you can draw it like how I showed you. Let us recall what are the parts. What is the stalk of the ovule called? Funicle. The point where the stalk joins the body of the ovule, hilum. Then what is the tissue inside the ovule the main tissue called Newillas, and inside the ovule there is a tissue, there is a generation called the embryo sac, a mass of cells which has egg in it, the embryo sac. So here they have shaded the embryo sac in green over here, you can see, and the surrounding tissue, obviously you can imagine there are cells in the surrounding tissue, and that is what is referred to as the Newillas. Then what are these coverings? See, this is the inner integument, this is the outer integument. These are the protective coverings which encircle the Newillas. And what do you call this point where the integument is not there? This is called the micropyle. Like I told you, the micropyle is the region which is called the opening of the ovule. And what is missing in the opening of the ovule? The integument is missing. Okay, the same diagram which I drew, which is shown here in an inverted or an upside down manner. So I hope the concept is clear to all of you. Now we had seen the structure of an anatropus. This diagram is a very important 5-mark question. Along with the explanation, you have to draw this diagram, but don't leave the embryo sac blank like I have shown here. As I continue with this lecture, we will fill up what is inside the embryo sac also. When you're drawing the diagram in the board exam, make sure you don't shade the embryo sac like I have shown here, but you should show cells inside that, which we will see how it looks like in the further slides in this lecture. Okay, so this is the structure of a megasporangium or the ovule. So what did we see? We saw the funicle, the point of attachment, hilum, the main tissue, Newillas, the protective coverings. How many integuments were there? 2 integuments, inner integument, outer integument. Where there was no integument, that region is called the micropyle. The base of the ovule was called chalaza. Sitting inside the ovule in the Newillas is the embryo sac, which represents the female gametophyte, and this is called a female gametophyte because, like I told you, the embryo sac contains the haploid female gamete, which is the egg inside it. Okay, now let us see what happens inside the ovule, or how is the embryo sac formed. So initially the embryo sac is not there inside the ovule. Okay, so we will be discussing about a process which is referred to as a megasporogenesis, which is happening inside the ovule. So I will not be drawing the entire ovule, but I will only be showing you a portion of the Newillas and the integument, and you will be able to understand what is happening. Okay, so what happens is, in the early stages of the ovule, so let me take this, the Newillas here. Okay, and there is a inner covering of the Newillas. This is the inner integument, and this is the outer integument. Okay, and I told you that the Newillas is a nutritive tissue. So let me show you some cells to make sure that there are cells which make up the Newillas. I told you the main body of the ovule is made up of the Newillas, which is nutritive in nature. So whatever cells I am showing you here, they are all the nuclear tissue. Okay, now what you must focus on is, in the Newillas there will be one cell which will differentiate itself, which will become different from all the other cells in the Newillas. This one cell, remember, there is no embryo sac still. We are learning the development of the embryo sac here. But the first step in the development of embryo sac is a process that I am showing you here, which is called megasporogenesis. Okay, so inside the Newillas, which is the micropyle? This is the micropyle, because here integuments are not there. And which is the opposite side of the ovule? The opposite side of the ovule, if you remember, we had called it as the chalaza. So this is the chalazal end, and this is the micropylar end. Usually what happens is, let me label this part. What is this tissue called? This tissue is called the Newillas. So usually the body or the Newillas of the ovule differentiates one single cell in the micropylar end. Now this single cell that is differentiating, that becomes different from all the other Newillas cells, will come to be known as, will function as a cell called the megaspore mother cell. It is diploid. Like every other nuclear cell, it is diploid. Remember, the nucleus is also a tissue made up of diploid cells. This much is clear to all of you. Where is this megaspore mother cell differentiating? The megaspore mother cell is differentiating at the micropylar end of the nucleus. Micropyle is the region where integuments are missing. What are these two coverings? The outer covering and inner covering of the nucleus. In the previous diagram we saw these were referred to as the integuments. So let me label the integuments also. There is an outer integument and an inner integument. This much is clear to all of you. Now what will happen is this megaspore mother cell which has differentiated will now undergo a process called meiosis. You all know that meiosis happens in two stages. I am writing at the top of the slide here. Meiosis is a reductional division where diploid cells become haploid, and meiosis is occurring in two stages, that is meiosis 1 and meiosis 2. Meiosis 1 results in two daughter cells. Each of the two daughter cells will undergo meiosis 2 to form a set of 4 haploid daughter cells. Okay, so now this particular cell, which is the megaspore mother cell, will first undergo meiosis 1. Now let us see what happens. I am again going to draw the nucleus and the integuments. In reality, the integuments are also developing. While this process is happening, the integuments are still developing, but here I am showing it complete so that you get an idea of where this process is happening. Once again, let us mark the poles. This is the chalazal end and this is the micropylar end. Let me call it as M. Okay, and what is this tissue I am drawing inside the ovule? I kept telling you that the main nutritive tissue in the ovule is, which is with lot of reserve food material, is referred to as the nucleus. So I am showing you the nucleus here, a part of the nucleus. Okay, so again let me label the parts. This is outer integument and inner integument. Okay, and this is the nucleus. Now this here there was only one megaspore mother cell, but now notice there will be two cells which are formed. Now what are these two cells? These two cells are product of what? They are a product of meiosis 1. So will they be haploid or diploid? Obviously they will be haploid. So these two cells which are sitting here inside the nucleus now, which are because of the meiosis 1 of megaspore mother cell, we call them as, we just call them as a dyad. Okay, dyad means what? A group of two. And how did they come? They came from the meiosis 1 of whom? Meiosis 1 of megaspore mother cell. Okay, now this is not the end of meiosis, because please remember both of these cells are now going to participate in meiosis 2. Both these, remember meiosis happens in 1 and 2 stages, isn't it? So once again let me draw the nucleus for you here. Once again the inner integument, the outer integument, a few cells let me show you which are the cells of the nucleus. Yes, but now there are 4 cells. 1, 2, 3, 4. How did 2 become 4? Because they underwent meiosis 2. Each one underwent meiosis 2. So now what will you call this group of 4 cells? Now these are called the tetrad. They are a group of 4. And these haploid cells are now called megaspores. A tetrad of haploid megaspores. Clear to all of you? So we started off with one cell. Which was that cell? We started off with a megaspore mother cell. A megaspore mother cell gets differentiated from the nucellus on which side? On the micropolar end of the ovule. Then it underwent what? Meiosis 1 to form a set of two cells. We didn't call them megaspores yet. These two cells will now undergo meiosis 2 to form a set of four cells. What are these haploid cells? Why are they haploid? Because they are product of meiosis. And what are these cells now referred to as? These cells are now referred to as what? Megaspores. And one very, very important thing is, out of these four megaspores, three megaspores will die or degenerate. If three of them die, then how many of them will survive? One surviving, the one who gets to survive out of the four is called the one functional megaspore. How many megaspores are formed? 4 megaspores. Out of those 4 megaspores, who will die? 3 of them will die. Only one survives. Who is that lucky one who is going to survive? Let us see this third diagram again. This is the micropylar end. This is the chalazal end. Yes or no? Micropyle is the opening of the ovule where integuments are missing. Okay. Now, among these four, only the one that is present near the chalazal region, this one, this lucky one, is going to survive. Whereas the other three, which are sitting below near the micropylar end, they are going to degenerate. So this is the only megaspore which I am going to label now, which is going to live. So what will we call this megaspore now? This is called the functional megaspore. This process, this entire process where I spoke of, what did we start with? We started with MMC. Let me call Megaspore mother cell as MMC. Megaspore mother cell, I will write it here for you. See the top of the slide. Megaspore mother cell. Was it haploid or diploid? Diploid. It underwent what division? Meiosis. To form what? Haploid megaspores. Totally how many? 4. But how many will die? 3 of them will die. So I'm going to write here also. These 3 of them, they are the ones who degenerate. The one at the chalazal end will survive. What do we call that surviving megaspore? We call it as the functional megaspore. This process where the megaspore mother cell undergoes meiosis to form megaspores, haploid megaspores, is referred to as megasporogenesis. The name itself has the meaning megasporogenesis, genesis of megaspore from megaspore mother cell. Clear? So one megaspore mother cell which came from the nucellus underwent meiosis 1 and then it underwent meiosis 2 to form a set of first a dyad and then a tetrad. Now look at the bottom of the page. Here all of you look at this bottom of the page here. Out of the 4, 3 will degenerate and only one survives. Please remember, where is the surviving megaspore usually? In the chalazal side or the chalazal end. Did you understand? Okay. Now let me again draw the nucellus. This has a lot of diagrams and it will be ideal if you also draw along with me. So nucellus, what is this covering I am showing? Inner covering, outer covering. What are these coverings? Integument of the ovule. Now what are these cells? Tell me, what is this body of nutritive tissue? Nucellus. Now how many megaspores are formed? 1. 2, 3, 4. Which one is going to live? This one is going to live. So what is this megaspore which is going to survive? Functional megaspore. The other 3 are going to die out. Now the functional megaspore is haploid or diploid? Haploid. Why? It is a product of meiosis. Now which is the micropylar end? This is the micropylar end. And which is the chalazal end? The opposite side is the chalazal end. Now this megaspore is going to live. Now let us see what journey is this megaspore going to take. Let me magnify this megaspore here for you. This is the megaspore that lives. So what did we call it? Functional. Haploid megaspore. There is only one megaspore because 3 out of 4 died. So this one megaspore, its fate is it has to develop to become the embryo sac. Embryo sac is also called female gametophyte. Why? It has egg in it. That's why. It's a plant like stage. It's a stage with it's called a plant, but it's very, very small with limited number of cells. So this one megaspore, the one single megaspore, develops into an embryo sac or female gametophyte. Its fate is, its goal in life is to give rise to an embryo sac. This kind of development where only one megaspore develops into the embryo sac is called monospory or monosporic development. Tell me why it is called monosporic development? Because do all the 4 megaspores become embryo sac? No. See this one that is surviving. Only one that survives is the one that is going to become, that is going to develop into embryo sac. Mono, the name only has meaning. In biology, everything is self-explanatory. Monosporic. Mono means one. Sporic means one functional megaspore that lives. See, this is the megaspore that is living. Others are dying out. This one develops into the embryo sac. Such a process where one megaspore forms one embryo sac is called I'm repeating this again and again because this is a very important 1-mark question. Why is the development of embryo sac in most angiosperms considered monosporic? Because they come from, the embryo sac develops from only one functional megaspore. Okay, now let us see how the development happens. So I am going to take an arrow from here and divert your attention to the top of the slide. Let us make judicious use of the space available. So this megaspore is now going to undergo a series of 3 mitotic divisions, but only the nucleus divides. So let us see what happens. So now this megaspore, it has become slightly bigger in size. It has 2 nuclei now. See, it had 1 nucleus. How did it become 2 nuclei? Because it underwent first mitotic division. But what is so strange about this mitotic division? Cell is still 1. Only the nuclei have doubled. So we call such a mitotic division as free nuclear division. When I say free nuclear division, cell is not, the cytoplasm is not dividing. Only the nucleus, karyokinesis is happening, cytokinesis is not happening. So first mitotic free nuclear division, we get how many nuclei? We get a megaspore with now 2 nuclei. Okay, now next. Again, a second mitotic division. which is also free nuclear. So 2 nuclei now become, see the cell is elongating. 2 become 4. 2 on one side, they start moving away from each other. See how these two went away from each other? The 2 nuclei are opposite, occupying opposite poles. Similarly, the 4 nuclei that are formed by the second mitotic division, 2 of the nuclei occupy one pole and 2 of the other nuclei occupy the other pole. The cell is also elongating. Okay, now this is not the end. So second mitotic division happened. Now what stage is it? 4 nuclear stage, 4 nucleate. There are totally 4 nuclei now. Now what happens is, as you can expect, a third mitotic division. I am not repeating again, but please bear in mind, how is this mitosis different from all the other mitosis? Because only the nucleus division is happening. Only the karyokinesis is happening. So now what do you expect? You will expect the cell has become even more longer, now elongated into a spindle shaped cell. Now 4 will become 8. So 4 becomes 8 nuclei. 4 nuclei becomes 8 nuclei, and see how all the 8 nuclei are occupying the opposite sides. The dots which I am drawing here represent nuclear envelope. So it's as if a single cell has become massive and now it has 8 nuclei in it. So let me call this as 8 nuclei or 8 nucleate stage. See how the cell is dividing, but not by traditional conventional mitosis. This is a mitosis where what division happens? Only karyokinesis, free nuclear. Okay, now out of these 8 nuclei, 2 of them will come towards the center. I'm sorry. Yeah. So 2 of them will move towards the center. So this one is coming to the center and this one is coming to the center. Otherwise, how many are there now? 3 are there. So I'm going to show now divisions are over, okay? So no more mitotic divisions. The cell has become elongated now. 3 are in the one pole, 1, 2 and 3. 3 are in this pole and only 2 have come to the center. Okay, now what happens is these 2 nuclei which come and occupy the central position are now called the polar nuclei. So please bear in mind, out of 8, 3 and 3 will remain here. 3 is remaining in this pole, 3 is remaining in this pole, 2 are coming to the center, called polar nuclei. Now very soon, around the 3 nuclei which are present in each of the poles, cell wall will get deposited. So again I am going to show the massive cell now. And I told you 2 nuclei had come and occupied the center. What are these 2 nuclei which come to the center called? They are called the polar nuclei. Okay. Now there were 3 and 3 in each pole. Which is the micropolar end and which is the chalazal end? See, this is the cell. This was the micropylar end and this was the chalazal end. Okay. So now the 3 nuclei that are present over here, 1, 2, they will organize into cells. See, 1, 2, 3. I told you cell wall is laid, cell membrane and cell wall gets deposited. So now they are no longer nuclei, they become cells. Here also 1, 2, 3. There were 3 nuclei in the chalazal end. Okay, and this cell in the center, this massive cell also will develop a barrier between these cells. See? it also develops a membrane. So now we have, let us count the number of cells now. 3 cells over this side, 3 cells on this side, and one massive cell in the center. So 3 plus 1 plus 3, we have 7 cells. Let us count the number of nucleus now. 1, so let me count here, 3 nuclei here, 3 nuclei on this side, and 2 in the center. So how many nuclei? 3 cell, 8 nucleate. Now this structure which has 7 cell and 8 nucleate. This structure which now has 7 cells and 8 nuclei is what is referred to as the embryo sac. I have not named the cells yet. I will come to the naming later. But this is clear to all of you, right? How a single functional megaspore, look here, a tiny megaspore became a massive cell, and an organization which has how many cells and how many nuclei? 7 cells and 8 nuclei. This 7-celled 8 nucleate structure is called embryo sac. Did you understand what I am saying? So what is this that we discussed here? The fate of the functional megaspore. The functional megaspore becomes the embryo sac because it is only one surviving megaspore that forms the embryo sac. We call the development as monosporic development. And how did it divide? It did not divide by cytokinesis. It only underwent karyokinesis. First karyokinesis, second mitosis or karyokinesis, third mitosis or karyokinesis, to become what? 8-celled stage. 8 nuclear stage. 2 of the nuclei migrate to the center, they become polar nuclei. They remain as nuclei only. The 3 on each pole, they become cells. So 3 cells in one pole, 3 cells in the other pole, and the central large cell becomes a cell with 2 nuclei. Totally 7 cells. How many nuclei? 3 plus 3 plus 2 in the center, 8 nucleate. Is that clear? Okay. So let us see the organization again. How many nuclei did we get? This is the huge cell now. I told you 3 nuclei will remain in one pole. Which pole? The chalazal end facing the chalaza in the ovule. Remember, this is happening inside the ovule, inside the nucellus. Then 3 of them remain in one of the pole, that is the micropylar end, and 2 nuclei will come to the center. And what is formed? Between the 3 nuclei, boundaries are formed here. See, they become cells, and these also become cells. Okay, and this, the nuclei which are present in the center as it is, they are the polar nuclei, and this massive cell will also develop a cell wall of its own, and it gets separated from the 3 cells at the chalazal end and the 3 cells at the micropylar end. And this, now let us name these cells. This massive cell is now called the central cell. And I'm going to write in brackets binucleate. The group of 3 cells which are present in the chalazal end are now called antipodals. The group of 3 cells which are present at the micropylar end is now called the egg apparatus. What is this egg apparatus made up of? Let me magnify the egg apparatus for you. In the micropylar end, you can notice 2 cells which are sitting next to each other like this with tapering ends, and one cell is protruding from between them. It's peeking out. isn't it? It's peeping. These two cells which are present on the sides and one cell which is present above, actually behind, but it is peeping out. These two cells which are present, actually it's a group of three, that's why it is called egg apparatus. These two cells are called the synergids. And the egg is this cell. So this is the most important cell which is protected by the synergids. See, synergids, it's sitting cozily between the synergids, peeping out from behind. Correct? And at the lower ends of the synergid, you can see certain infoldings of the cell wall, like this. Finger-like projections of the cell wall. This infolding of the cell wall is called the filiform apparatus. It plays a very important role in fertilization. We will see later how it plays an important role in fertilization. Okay, so the egg and two synergids together constitutes the egg apparatus. Yes or no? Egg apparatus which has two synergids and one egg. At the lower ends, where the synergid is attached to the embryo sac, the wall of the embryo sac, there are filiform apparatus which are infoldings of the cell wall of the synergids. And which is the largest cell of the embryo sac? It is the central cell. It has how many nuclei? 2 nuclei. Clear? So now you saw the organization is a 7-celled, very, very important, 8 nucleate structure. The embryo sac or the female gametophyte is a 7-celled, 8 nucleate structure. It came from where did it come? How did the embryo sac come? Did it come from all the 4 megaspores or one functional megaspore? This is also a revision point for you. One which survived near the chalazal end, functional megaspore, gave rise to the embryo sac. How many mitotic divisions? 3. Cellular or free nuclear? Free nuclear, karyokinesis, no cytokinesis. Later the cell wall develops, see, so that it becomes how many celled structure? 7-celled structure. And what is this development called where one functional megaspore develops into the embryo sac? Monosporic development or monospory. Okay? Now how you should draw the ovule actually in the examination for a 5-mark question. Okay, so you should show the embryo sac inside. So let us draw the ovule. Placenta, I told you there is a nutritive tissue. Then this is the stalk and this is the body of the ovule. And these are the protective coverings. This is the outer membrane. What is the outer covering called? Outer integument. And this is the inner integument. Correct? Now let us show the Newillas inside. Inside the Newillas, let us show the embryo sac. Now you saw the embryo sac, how it developed from a megaspore. Okay? So show cells surrounding the embryo sac, which is the Newillas. Yes or no? So I am filling up the gap. What is this tissue which is surrounding the embryo sac? This is the nutritive tissue, the main tissue of the ovule that I was talking about. What is this main tissue called? The Newillas. So it is time consuming, but it will fetch you good marks if you are patient enough and if you fill up all these cells the body of cells which are sitting inside the ovule. Okay? Okay, so Newillas is done. Now embryo sac. 3 cells, which is the chalaza. Before that, let us label the stalk. The stalk. This is the stalk. Let me put an arrow like this. This is the, what did we call the stalk? Funicle. Very good. Now the point where the stalk, see this arrow which I am showing here. See this line of the funicle. The funicle is joining the body of the ovule. This point where the funicle joins the body, the junction between the funicle and the body, hilum. The base of the ovule, chalaza. The 2 coverings of the ovule, outer layer, inner layer. What do we call these protective coverings? We call them as integuments. Yes or no? Okay. Then what is the main tissue here? Let me label this main nutritive tissue with reserve food material, from which cell we got a megaspore which became the embryo sac. Newillas, the most important part of the ovule, the body of the ovule. Newillas. Now sitting inside the Newillas, it is the embryo sac. Before that, what is this region called where integument is absent? Micropyle. The opening of the ovule. So in brackets I am writing opening of ovule. Now let us draw the embryo sac. 3 cells at the chalazal end. 1, 2, 3. What should I label them? Now you don't have to label the cells in the exam. It's better if you do. These cells are called antipodals. Then, what are the 2 nuclei in the center? I told you 2 nuclei migrate to the center. They may or may not fuse later. During fertilization, they will obviously, in some, before they remain as 2 nuclei. So, what is this cell? The large cell which has the 2 nuclei. Central cell. Yes or no? And what are these nuclei? Polar nuclei. Okay. Then there are group of, I'm so sorry. Okay, I'll have to draw it all over again for you now. It's okay, it doesn't matter. Okay, now I am not going to label all the parts. Okay, let me come to embryo sac. You can rewind and see what are the parts. I'm just showing the ovule, the protective coverings of the ovule. I hope you know what I'm drawing now. Integument, funicle, the point where the funicle attaches, hilum, then the massive tissue inside, Newillas, and I was showing you the embryo sac. So let me come directly to the embryo sac now. Okay, so I told you that surrounding the embryo sac, what is this nutritive tissue? What is this I'm filling up here? Newillas. You fill up the gaps in your books. In the previous diagram I had filled it up. Okay? So this is the Newillas. Okay. Then I told you antipodals. I'm showing you the embryo sac now. What did we label these three cells? Antipodals. Then the cell, the large cell in the center, central cell. It has two nuclei. What are the two nuclei? The polar nuclei. Then, which is the chalaza? This is the chalazal end. We had labeled this as the chalaza of the ovule. This opening where there is integument is not there. What did we label this? Micropyle. The stalk, funicle. The junction between the stalk and the body, hilum. Correct? Then what are these layers? Now this nutritive tissue I had labeled on this side only. So I label it this side. Newillas. And these coverings of the ovule which cover the Newillas. Integuments. So this is how your 5 mark should be. The diagram should be. Integuments. Now let us continue with the embryo sac. We finished labeling the antipodals. See, 3 cells at the chalazal end of the embryo sac. Now let us label 3 cells this side. Let us draw 2 cells and 1 cell peeping out. What is this cell that is peeping out? Egg. Haploid. What are these 2 cells which are surrounding the egg? Synergid. I am labeling 1. So let me call it as synergid. But remember there are 2 synergids and antipodal is 3. What do we call these 2 together? The egg and the synergid. The egg, apparatus. This entire thing is now called the embryo sac. What is the alternate term for embryo sac? I told you. Female gametophyte. Why is it called a female gametophyte? Because it has what? Egg in it. So this is how the diagram for the ovule or the megasporangium must be represented in your examination. You must show the embryo sac sitting inside. Ideally, you must label, don't leave it blank in the exam, because in the textbook it is left blank. Of course, they have dealt with it later, but in your examination, you have to show each and every part of the embryo sac that is sitting inside the ovule. And like I have shown here, you also have to show the nutritive tissue. I'm continuously calling it nutritive tissue, but remember it is the main tissue of the ovule, which is referred to as the nucellus. See, you should show lot of cells in the nucellus. And sitting in the center of the nucellus, you have to show the embryo sac. So what is this embryo sac? I'm writing it again for you here. How many cells, how many nuclei? 7 cells, 8 nuclei. 7-celled, 8 nucleated structure is the embryo sac. Is it clear? So I hope the structure of the ovule and how the embryo sac came, that is also clear to all of you now, that it came through megaspore. Megaspore was formed first. There was nothing before. When we saw before, there was no embryo sac inside, only nucellus was there. In the nucellus, I told you, one cell becomes different from the others. It becomes the megaspore mother cell. That gives rise to 4 megaspores. Only one survives, and that surviving megaspore becomes the embryo sac, which has the egg inside it. Okay, I hope you have understood the concept. So now coming to a quick recap of what all we learnt. Haploid 4 megaspores are formed from diploid megaspore mother cell. What is this process called? Megasporogenesis. How did the megaspore come? A single cell near the Newillas towards the micropyle. It became what? It became a megaspore mother cell which underwent, do you remember what division it underwent? Meiosis, to form 4 haploid megaspores. How many of the 4 die? 3 of them die. Only one megaspore survives, which is called the functional megaspore. What happens to that single megaspore that is living? It becomes the embryo sac. Because there is only one megaspore that becomes the embryo sac, what is this development called? Monosporic. Then it undergoes free nuclear divisions. First it becomes, the surviving megaspore becomes 2 nucleate. Then after 2 nucleate it becomes 4 nucleate. Then after 4 nucleate it becomes 8 nucleate. And the 8 nucleate structure organizes itself into a 7-celled 8 nucleate stage. The 7-celled 8 nucleate stage is nothing but the female gametophyte or the embryo sac. Yes or no? 7 cells, 3 in the chalazal end, antipodals, 3 in the micropylar end, egg apparatus, and 1 in the center, the largest cell. Which is the largest cell? The central cell, which has 2 nuclei called polar nuclei. Okay? See, in the textbook they have given this diagram. So this is the single cell which becomes differentiated from the nucellus of the ovule at the micropylar end. And see, I told you dyad is formed first. Why dyad? Because first it undergoes meiosis 1, and then each of them undergo meiosis 2, and then they become what? A megaspore tetrad. Out of these tetrad, how many will die? 3 will degenerate or die, and 1 will survive. And this is that one cell which is the functional megaspore. So see, it becomes 2 nucleate, 4 nucleate, 8 nucleate. 2 of them come to the center, they become the polar nuclei. The 3 organize. Now this is the micropylar end, and this is the chalazal end. Towards the chalazal end, so here it is completely reversed. Suddenly they reverse the pole. See here, this suddenly becomes the chalazal end, and this suddenly becomes the micropylar end. Until this diagram, it is, they follow, micropyle is on the up and chalaza is below. So that's why I gave you the other diagram for the ovule, which is given upside down in the textbook. Okay, you can draw it however you want, but don't get confused which is the chalazal end and which is the micropylar end. In the chalazal end, you can see antipodals, 3 antipodals. The largest cell, this cell is called central cell. It has 2 polar nuclei. Now see how the egg is peeping out, and then these are the 2 synergids. Together they become the egg apparatus. And I told you that there are cell wall foldings in the synergid, which is called the filiform apparatus. So this is how a typical embryo sac looks like. I hope you all know that it is a 7-celled 8 nucleate structure. It's called an embryo sac, or it is called what? A female gametophyte. So what did we learn today? We learnt gynecium, we learnt the component called pistil. We saw that if the pistil is free, they're apocarpous. If they're fused, how do you know they're fused? You look for the stigma. More than one stigma will be there. Or you take section of the ovary and look for the compartments called locules. Then you will know that they are syncarpous. Then we saw the structure of megasporangium or ovule. Then we saw how the haploid megaspore is produced from MMC. Go and find out what is MMC. That is called megasporogenesis. Then we saw that one megaspore becomes an embryo sac or a female gametophyte. And then we saw the structural organization of the embryo sac. Time and again I have been repeating, 7-celled 8 nucleate structure of the embryo sac, which has what? Which has the egg in it. So for homework, you will describe the structure of, I told you that the ovule is bent on itself 180 degrees, it is turned, and that's why it is called anatropous ovule, isn't it? So that is what is anatropous ovule. The diagram which I drew, the diagram given in the textbook, but it is given upside down in the textbook, or in some plants it may be organized like that, but we, for our convenience, we consider the classical diagram which is given in most of the books. So that is the anatropous ovule. You have to and you know that the ovule is also called megasporangium. You have to describe, plus you have to draw a neat label diagram. I told you, draw the diagram with the embryo sac inside. Don't leave the embryo sac blank. Explain the process of megasporogenesis. How is a haploid megaspore formed from a diploid megaspore mother cell? Describe the process of formation of the 7-celled 8-nucleate embryo sac from the one megaspore that lives, that survives out of 4, and that is for a 5-mark question, along with the diagram showing how the nuclei divide and how it becomes a 7-celled 8-nucleate structure. Okay, so with this we complete the lecture 2 of sexual reproduction in flowering plants. Thank you.